I apologize, it took me a little bit longer to post the grades than usual. I had a funeral on Wednesday morning, and that kind of obviously took time to do things. I've taken a look at the exams now and reflected on the distribution of grades and which questions were what and what the pattern was and so forth. And it seems to me, having looked at all of those, that there were some that were more difficult than were intended, and a couple where a quote wrong answer was chosen by students more often than the right one, but if I'd been able to offer partial credit, I could have offered you partial credit for it. The computer doesn't allow that kind of thing, and so since I don't have the option to tweak it to that degree, what I've done is that for this test, as for the last ones, the thresholds have come down by come on, this thing is, there we go. One of the slowest systems I've ever dealt with. At any rate, here we go. And I can unmute this image now, and text size, largest. So here, this should be visible, I think. This is the thresholds for this exam. It isn't a problem with the multiple choices. I mean, there are some questions where, you have A and C, B and D, that kind of thing. And some of those questions were the ones that 90% or more of the students got right. So it isn't simply the structure of the questions. It was certain particular topics, and I'll keep working on trying to teach those better. But I think this is probably the best way to deal with the whole thing. There were no questions that were clearly bad questions, where students who understood the rest of it consistently got the wrong answer. It was more a matter of being more difficult than intended and perhaps not as strong a question as they could have been. So I hope that will accommodate it. Are there any other questions about the third exam before we move on? Just a reminder that the fourth exam that will cover the last 4 chapters is coming up on us in about 2 weeks from Monday, and the final exam a week after that. So I do hope you're beginning your review of the course material for the final already. It isn't really something that can be delayed altogether, and staying current with the material we're doing now. If you've been challenged by the material in metabolism, be of good spirit. Molecular biology for some people is just simply easier to wrap their minds around than metabolism, and is sometimes the place where people can kind of catch their breath again and pull ahead. So still good things to come. Any other kind of course organization things before we return to the structure of DNA and its replication? Okay, open. All right. There we go. Okay, so let's go in. I think we had gotten up to, we were about to start on slide 39, I think. Is that correct? Does that, we were on 38? So if you take samples of our cells, anyone here in the room, typically around 50 cell divisions in culture. Someone who's 5 years old might have a few more. Someone who's 70 will have definitely less. And this is partly because even though telomerase is good, it's not perfect, and telomeres tend to be a bit shorter over time. The maintenance of them is not perfect. And once cells reach what's called senescence, they may stay alive for a very long time, but they won't divide again, at least in culture. And that may represent something in our bodies as well. Cells that are immortal in cell culture, that is, that can be passed again and again and again, typically these are derived from cancer cells, but they can be derived in other ways. Cells like that typically have active telomerase enzyme activity. So they have abundant enough activity to keep the length of those telomeres out and long and bushy. Since the chromosomes have been maintained and are not slowly being eaten and broken up, the cells can continue not merely staying alive, but growing enough to replicate. And I think I read this one, find the key to immortality tomorrow. I got 'em up. So, let's go into the screen mode here. All right. So, and the telomerase is actually an extremely relevant enzyme for another reason. because it may well be related to an enzyme that we've met earlier in the course, HIV reverse transcriptase. HIV is human immunodeficiency virus, the causative agent behind AIDS, acquired immunodeficiency. And the enzyme is actually an interesting kind of structure. It's a protein that has two activities. So this would be a case where one stretch of DNA would code for two phenotypes, right? Because it has both the enzyme activity of being a polymerase, of generating more nucleotide, and it has a similar structure to the DNA polymerases we've looked at of the palm, thumb, fingers thing of being able to grab and pull in nucleotides and slap them onto the growing chain. But HIV reverse transcriptase also has an RNase activity, so it can degrade RNA molecules. It's unusual for polymerases in that it can use templates of either DNA or RNA. So although what it synthesizes is deoxyribonucleic acid, it can use templates of either. And then the RNase activity degrades the template. So if it started off from an RNA molecule that was in the virus, after 2 rounds of activity, what you now have is a double-stranded DNA molecule. And it's provocative because it looks like it may have come from the telomerase, which is able to synthesize DNA without having, by using that RNA template integral to the molecule. So, back. There we go. So this is one aspect of sort of the daily life of DNA. How do you make more of it when you need more of it? And that's what the polymerases accomplish. But a very significant problem is also DNA packaging. So again, just to bring it home to you, each cell has a meter of DNA packed into a nucleus that's maybe 10 microns across. So a real significant problem in packaging. And the way in which DNA is packaged turns out to have effects on many aspects of its activity. And this is an electron micrograph, a thin section transmission electron micrograph of an eukaryotic nucleus. So this is allowing us to look at a very thin slice of the nucleus in very high resolution. And what we're, this arrow is not well aligned. I'm sorry. This arrow should come off of the euchromatin. In fact, the text here has rearranged itself dramatically, and I'll tell you which arrows should be what. In broad terms, in an interphase nucleus, a nucleus that is not going through mitosis, in an interphase nucleus, the DNA can be roughly categorized into 2 big heaps, not perfectly, but with considerable consistency. There is some of the DNA is dense, is densely packed. It stains intensely. When we look at its activity, it turns out that it's not being transcribed to a great extent, and it typically is going to be found near the periphery of the nucleus. That's called heterochromatin. The other colored stuff is what the Greek means, is the other colored stuff. This arrow, which is correctly pointing to some euchromatin, points to the rest of the DNA. Euchromatin, the DNA is typically in a looser configuration. It's more accessible to enzymes that might degrade it. Most of the transcriptional activity will be happening in the euchromatin, and it will typically be towards the center of the nucleus. So the whole meter of DNA in your cells is going to be roughly divided between those 2 groups, and they differ in their chemical properties in ways that brings all that about. Now, one of the first levels of organization, the first thing that cells do to start making the DNA take up less space, is to pack it into what are called nucleosomes. Some little body, nucleus in the nucleus. The nucleosome is a repeating structural unit for packing DNA into eukaryotic chromosomes. So you're not going to see this in the prokaryotes. This is something eukaryotes do. And it's a combination of the DNA itself and proteins that are specifically for this purpose, called histones. And you can see here a diagram of this kind of a structure where you have 8 monomers here as the core, the DNA wrapping around it in loops, and another molecule of histone sort of anchoring that thing together. So this is the first level of packaging that's going to accomplish this task of putting that meter of DNA into it. The presence of these nucleosomes is also one of the things that turns out to be playing one of the roles in modulating transcription, whether or not a sequence of DNA at any given instant is going to be used to make RNA or not. The nucleosome is sometimes called beads on a string. That is, when you have some DNA and histones and that's all you have, you can look at that on an electron micrograph and see what looks like little beads on a string. And what that's the result of is seeing little dense patches where you've got the histones and DNA and a slender strand between them that's just the DNA itself. The core here is composed of 8 proteins, 2 copies each of H2A and H2B, 2 of H3, 2 of H4. This is showing you the structure at the ribbon diagram level of the nucleosome core, this octamer. And then the DNA goes around the outside of these. So the histone core is like the spool when you have a spool of thread, and then the thread goes around the spool. The histone tails are able to interact with the DNA. And so in this drawing, you can see here's one histone tail, and here's another histone tail, and there. So that the tails of these proteins are capable of interacting with that backbone of the DNA that's out there. Here's a space-filling diagram of a nucleosome. You can see, again, the double helix of the DNA wrapped twice around that central core. This is a little higher resolution one. Here you can see the blue here are the H3 molecules. The green are the H4 molecules. The orange is H2A. The red is H2B. And perhaps more importantly, these tails are a little more visible here as being flopping out, capable of sort of extending from the core to touch in appropriate parts of the DNA molecule. If we look at this in a little more specific detail, it turns out that you can wrap 146 base pairs around that 8 histone core protein. If all you have is those 8 histone core proteins, it's a looser, floppier structure. H1, or histone 1, crosses over the DNA that's bound around it to sort of staple it in place, if you like to think of it that way. And that provides for further compaction as well. Nucleosomes by themselves can give us about 30 to 40-fold denser packing than we have in the absence of any proteins. We're not there yet, but we're closer. The next level of organization that gives further compaction is if you have these nucleosomes on DNA and then you, in effect, twist that, you're going to start to get coiling of that beads on a string structure, and you end up with a structure that looks something like this, called a solenoid. Have any of you seen a physical solenoid? Could you raise your hand? Any of you have? Oh, good. Motorcycle? People who actually handle real engines may see real solenoids. They involve wrapping coils of things, and it's an old-fashioned way of generating sparks and so on. But in effect, this is a coil of the coils, and can pretty easily, you can see, would compact this quite a bit further. And compaction into this kind of solenoid is going to reduce the chance that it's going to get tangled from flopping around the nucleus. It allows the nucleus to locate it where it wants to be. But it may restrict access to that DNA for things like replication or for transcription. So the solenoid is a characteristic feature of mitotic chromosomes, but perhaps not as characteristic in the interphase nucleus. And this is a chance to see these solenoids physically. These now are electron micrographs that are done by negative staining. You have a sample in solution. You mix it with heavy metal salts, spread it on a thin film, and put it in an electron micrograph and shine light through it, or electrons through it, sorry. This kind of wavy, loose stuff is more like a typical beads on a string structure. But you can modulate the extent to which those things pack together or not by changing salt concentrations, whether or not H1 is present, whether the histones have covalent modifications on them, things like that. The degree of supercoiling of the DNA, so if you have a plasmid DNA that's a circular molecule and you're adding histones to it, and then you change the degree of supercoiling, you may twist it into a solenoid or out of a solenoid. And you can see here, for instance, a stretch where that solenoid structure is forming and is much more compact than the simple beads on a string structure. Hi, do you have something to add to the conversation? Okay. Yeah. All right. I think it kind of boggles the mind a little bit to think about what the DNA is doing in these solenoids. Since the DNA travels around the outside of the histone, so around each histone you've got the two loops of DNA, but in the solenoid you're kind of taking this curving path. It's a very indirect pathway for the DNA to be following, and so you can understand that it's going to be harder to get access to that DNA. It also reduces access to the surface of the histones. You can see that part of the surface area of the histone core is buried from the fact of being inside the solenoid. This is a schematic that shows you the other levels of packing, or the other higher orders of packing. Here's your naked DNA, just a double helix and 2 nanometers across. In a beads on a string situation where all you have is the nucleosome cores, maybe 11 nanometers across. So we're now a little bit thicker than an actin filament. If we then pack that into a solenoid, this is sometimes called the 30 nanometer fiber because it's 30 nanometers across. This compacted form is then formed into loops. So the bases here are attached on some kind of a structure that is making the chromosome a chromosome, and these loops are sticking out of it kind of like the bristles on a bottle brush. Although the DNA, if you follow it, is one continuous molecule of DNA. What this packing does is it allows the cell to treat regions of that DNA as semi-autonomous domains of DNA. And the diameter at this point would be more like 300 nanometers, about 10 times as far across. And then this, in turn, is a fiber sticking off of the chromosome. So you can see that there's several orders of packing of the packing of the packing. And the consequence is that you get 50,000-fold shorter space needed to hold the DNA. And you've got multiple chromosomes, which takes care of the rest of it. The exact degree of packing, the density of these coils, the fraction that are in solenoids, things like that, are altered by a number of different things. Histones can be modified. They can have covalent modifications to the proteins themselves. There can be histone remodeling, where different other factors interacting with them can spread them out or move them closer together. And then you can actually have different proteins in there. That is, there are the core histones that are the usual ones, but there are some variant histones that are only found in certain limited circumstances that allow for other tweaking to go on. I've mentioned a couple of times now the idea of histone modifications, and I mentioned that the tails of the histones stick out from the core and can interact with the DNA. The histone tails are very flexible, and this is showing you, here's your histone core, your tube of DNA wrapped around it, and the various tails sort of flapping in the breeze. And you can see they've put little dots for all the different sites along it that are potential sites for modification. So if you look at all those dots, you can see that any given histone core has a very large number of possible combinations of modifications. And this is some of the kinds of things that can happen to it. They can be acetylated. You can add a 2-carbon fragment to it. You can methylate it, make it more hydrophobic. You can ubiquitinate it. Ubiquitin is a small, highly conserved protein. We're going to meet it again when we talk about protein degradation. Sumoylation. Sumo is a protein that has some homology to ubiquitin. phosphorylation, that would make it more highly charged. So you see you've got a really wide array of modifications you can do to these histone tails to make them everything from much more hydrophobic to much bulkier to more charged. You can really tweak the physical properties of the histone core by these modifications. Sometimes this is spoken of as the histone code. I don't know if the readout of these modifications is as exact as the genetic code, but the histone code is proposed as a way of thinking about how you can use the same gene sequence in different ways in different cells. Your neurons and your muscles and your liver cells have the exact same DNA in them, but they're doing very different things. And part of why they're doing different things is their histones are modified in different ways on different genes. And the idea that these modifications are part of the basis for those changes in expression is what's meant by the histone code. So to give you some examples, you can acetylate lysine. So you can go from a positive charge there to an acetyl group, which is neutral. You can methylate lysine, so you can cover it up with something hydrophobic. You can phosphorylate serines and threonines, so you can make them more charged. And all these little modifications then are potential sites of interaction with proteins that are involved in transcription that can recognize those modifications and then modify the DNA protein interactions. Questions? This is some of the, one of the more dramatic diagrams I've seen for this. Medusa turns into a pillbox hat. Two of the key enzymes that are involved in this are called histone acetyltransferase, or HAT, and it, surprisingly, transfers an acetyl group onto histones. So a histone acetyltransferase would tend to modify the positive charges into neutral ones and therefore open it up like this because the negative charges, the positive charges that were on the unmodified thing are going to be interrupted and released from the DNA backbone. And in general, but not always, a histone acetyltransferase will tend to produce a more open configuration of the chromatin. It tends to be associated with transcribed areas or turning a gene on for transcription. Histone deacetylase, again, remember our rule of biochemistry, if there's an on switch, there's an off switch. Histone deacetylase removes those acetyl groups, exposes the positive charges of the lysine. The positive charges on the lysine see all those phosphate backbones and slip back down again. Histone deacetylase tends to be associated with more compact DNA, which tends to be less transcribed, or we say silent. So it's not a perfect one-to-one matching, but there's a considerable correlation between these things. So acetylation tends to make an open configuration. Open configurations tend to be transcribed. Deacetylation tends to make a compact one. Compact ones are harder to transcribe and tend to be silent. Histone methylation is another important one. This is another active area of research. And so if here you have your lysine, the positive charge that can interact with that phosphate backbone. Here's what the acetyl lysine looks like. Here's what a trimethyl lysine looks like. There is still a positive charge buried in there, but it's got all those methyl groups sort of fending it off. And you can have anywhere from 1, 2, or 3 methyl groups added to the lysines. And there are also demethyl transferases, ones that take it back off again. Any given lysine residue might be a target for either methylation or for acetylation. And particular lysine residues are typically targets in particular tissues, so that there's a growing, careful, painful association of which residues are associated with which genes being expressed. We're a long way from there yet. I mentioned that there's also histone remodelers where you can change the spacing of histone cores, or that you can change the histone variants, that you might have different histone proteins put into there. And this is showing you an example of that. Here's the, this is sort of diagramming the three different things that we've talked about. Here you have a canonical nucleosome, the standard one we've talked about so far. Here's one that has different composition to it. Here's the canonical one that's been modified in various ways, like acetylation. And here's one where it's moved over to further down. H2AX is often replaces H2A if DNA is damaged and needs to be repaired. We can assume that that's involved with making that accessible to the repair machinery. So far we've talked about packaging of DNA or making it more or less available for transcription through the histones. That is, the DNA has not been touched, but the histones may be modified to make it more or less accessible. We also see changes of the DNA itself, chemical changes of the DNA itself. So there can be DNA modification. And one of the common ones is cytosine. Here's methylation on the cytosine. And this is one of the more common, so far as of the things that happens, this is one of the more common ones. I don't want to suggest that many cytosines are methylated, but that of the modified bases, many of them are methylcytosine. And you can see that it's in a position where it would stick into the major groove. I don't know if you remember back to the beginning of the course when I talked about the major and minor groove of DNA and how there were unsatisfied hydrogen bond donors and acceptors. Well, this methyl group on the cytosine points into the major groove and has the potential of changing the available binding sites for recognition by DNA binding proteins. Cytosines in particular that are next to a guanine, what's called a CpG sequence, are among the more common targets for methylation. And near the start of genes, they tend to be non-methylated. So there is some suggestion that methylation may be one of the ways that genes are turned down so they aren't active in tissues where they aren't needed. We know, for instance, that methylation of DNA is an important aspect of packaging the gametes so that the DNA in sperm and in oocytes is methylated in particular ways, to the extent that there are some traits that are different depending on whether you get your DNA from your mother or your father, because the mother or father methylate the DNA in different ways. So this can have quite major consequences for us. Now, a chemical modification is, a chemical modification. It's not something that's going to be replicated spontaneously during transcription unless something's done about that. And that turns out to be the object of machinery in its own right. That if we have here a stretch of DNA and there's particular cytosines that are methylated. And one thing about a CpG sequence is that you're going to have a pair of cytosines near each other, right? This is CpG here, methylated, and CpG over here, methylated. So this dyad here, or this dinucleotide, is the site of this modification. If all we do is replicate this DNA, we can end up with molecules in which only half of those sites would have the methyl group. And so there are enzymes that can recognize what's called hemimethylated DNA and add a methyl group across the strand to the other one. So this one had its methyl there, and the methylase added it across the strand. This one had it down there, and the methylase did there. So we've preserved the pattern of methylation so that you can have a stem cell, for instance, that has a methylation pattern and transmit that stably to the daughter cells, even though the gene sequence has not been changed. This is part of the basis for what I was just talking about called genomic imprinting, that some genes that you inherit will be more or less active depending on the parent you get them from, and this is mediated through gene imprinting. This can be especially dramatic in plants, where if you grow plants at high altitude for a while, they'll have a stunted body shape and small leaves and everything. And the seeds from those plants may go on producing small plants for quite a while. You may have to raise them down in Iowa for several generations before their full potential is released, because the methylation has to pass off over time. One of the other aspects of DNA modification is that it makes possible a particular form of mutation that would not be possible otherwise. If you have here a methylcytosine, that in the normal course of things, if it happens to deaminate, if it happens to lose its amine group spontaneously, what you've done is to create actually a molecule of thymine. And so this would now generate a different partner across the strand during replication. It's no longer something that's recognized by methylases. It's recognized by DNA polymerase as a thymine. And so at the next round of replication, it'll insert an A across the strand from it. So this is one of the sources of spontaneous mutations. Chromosomes, strictly speaking, the really highly organized, tightly packed thing, are things that we usually see at mitosis, when the cell is particularly challenged to separate DNA into exactly 2 equal halves. And during interphase, the time between mitosis, the organization of the chromosomes is looser and a little harder to summarize in some ways. You have the heterochromatin packed away at the edges and the euchromatin near the center, but it doesn't quite lend itself to the same kind of very pretty hierarchical organization of the metaphase chromosomes. But there are some interesting points where we see DNA packing during interphase. In mammals, sex determination is generated through chromosomes and which ones you have. So females have 2 X chromosomes, males have 1 X chromosome and 1 Y chromosome. The Y chromosome obviously doesn't contain something that's essential for life per se, or women would be dead. We aren't. It carries information for being male, but not for being alive. And so female mammals have these 2 X chromosomes, and they really only need 1. Men do just fine, or fairly well, with just 1. And well, they die younger. I mean, they don't do quite as well. And so what turns out is that female mammals will inactivate half of their X chromosomes. And this inactivation is random. It happens very early in development. And the inactivation is essentially a stable trait that is not going to change. So if we look at these cells, these are interphase cells, probably epithelial cells from the mouth or something like that. We'll notice a black, dense body off on one edge of the nucleus, there and there and there. And these are called bar bodies for the scientists who first described them. That in the average female who has 2 X chromosomes, her body cells in general will have one of those 2 X chromosomes inactivated in one of these little bar bodies off at the periphery of the nucleus. And the bar bodies are one of the ways that we can learn things about how things are inactivated, because they're such an abundant, explicit test case. And we find that the DNA in bar bodies is hypoacetylated. Remember I said that acetylation can help to open up the histone tails off of the DNA. So it's hypoacetylated, so it'll tend to pack down. The CPGs tend to be methylated. Remember, they're undermethylated near the active genes. They're methylated here. And there's a particular thing peculiar to bar bodies called X-ist RNA, which is one of the very few transcripts of the bar bodies. And it's an RNA whose role is to help compact and inactivate the rest of the chromosome. Bar bodies bring out some of the difficulties in defining what it means to be male and female. The shorthand rule of thumb is 2 X chromosomes female, an XY chromosome male, but there are other variations out there. So for example, someone who has Klinefelters has 2 X chromosomes and a Y chromosome. A Klinefelter's individual, in most cases, looks pretty much like an average guy. He may be a little taller and slenderer than usual, but a guy. But a man with Klinefelter's will have an X chromosome inactivated. He'll have bar bodies. So a bar body in this case is not a sign of being a woman. Some individuals have only one X chromosome and nothing else on the other side of it. These individuals are phenotypically females. They have an X chromosome, and so, you know, they handle those things. But they don't have one to spare. They have no bar body. So although they are phenotypically female, they have no bar body, which would be more characteristic of a man. And there are some individuals who are XX males who have had a gene rearrangement so that the necessary genes for male expression are now sitting on an X chromosome. These individuals don't inactivate that chromosome, and they have no bar body, but they have two X chromosomes. So at the genetic level, these things get a little bit murkier. There's the average rule, and then there's the various biological variations beyond that. I used to make comments about how would you make a law about the marriage of one man and one woman, but the Iowa State Supreme Court has made that a pointless question now, so we don't need to worry about it. It's still interesting biochemistry. The organization of chromosomes of DNA into chromosomes is sufficiently characteristic that we can actually identify individual chromosomes in human beings. They come in enough different sizes that you can physically separate them through flow cytometry and then identify sequences on them that allow you to make probes to them that are analogous to those sequences. And this is an example of what's called chromosome painting, where such probes have been marked with fluorescent probes that allow us to have different colors for different chromosomes, and you can now sort of look and see, you know, which of the various chromosomes you've got present. One of the values of this kind of chromosome painting is it makes it extremely easy, for example, to identify situations like this. Down here at the tip of this chromosome, and the tip of this chromosome, the tip of that chromosome, you can see that this individual has had rearrangements so that part of chromosome 13 is now over here on chromosome 6, part of chromosome 8 is down on the tip of 13, and part of chromosome 6 is over at the top of this chromosome 8. So this is an example of rearrangement, chromosomal rearrangement. This ability to paint individual chromosomes has allowed us to ask questions that we could not ask before, that were only possible once we could uniquely label one chromosome at a time. In this image, we have what's called a chromosome spread, where the nucleus has been smushed and all the chromosomes are spread out. You can see individual chromosomes, and you can see that only 2 chromosomes are painted with this particular probe. The others, the pale blue ones, are just showing all DNA. What happens in that case is something very interesting, which is that the chromosomes tend to have positions in the nucleus. If you had this mental image of the eukaryotic nucleus as kind of a bowl of soup with macaroni flowing around in it, it is far more structured than that. And individual chromosomes have particular and fairly compact locations within the nucleus during interphase. And that these locations vary with tissue type and they vary with the chromosome. So there's a level of organization even up to the level of the nucleus itself that we're only now really beginning to understand. Any questions about that? I'd like to sort of introduce you then at the end, I think this is our last topic on this, to something called the C-value paradox. And the C-value is the amount of DNA per haploid cell of an organism. So in us it would be in eggs or sperm, and in protozoa it might be in the whole organism. And the C-value paradox is that the amount of DNA you have is not a very good way to predict how complicated you are. And organisms that are similar to each other can have 100-fold differences in how much DNA they have. So if we look at this particular curve here, there is a very rough curve on this particular diagram of more complicated means more DNA. Because down here you have viruses and bacteria, and here you have echinoderms, and up here you have mammals and flowering plants. Flowering plants are more complicated than us in some respects because they can make a living on their own, and we can't. We depend on the plants. But if you look at any given evolutionary group, here's amphibians, for instance. This down here is a logarithmic scale. These amphibians are differing by over 100-fold in how much DNA they have. They're all amphibians. They all look like frogs, you know. Here's flowering plants. Now, flowering plants, admittedly, can vary quite a bit. You've got oaks and you've got duckweed, but still, this is like 3 orders of magnitude. So this was quite a challenge. I ran across this getting ready for the lecture, and it's sort of my current favorite illustration of the C-value paradox. The placozoan is a multicellular organism that has a top and a bottom, but no front or back or left or right. It's basically a pancake. And its bottom has cilia that it walks on, and its top is smooth, and it has mesenchymal cells. And if you cut it in half, it seals up, and you now have 2 trichoplaques. And if you turn a trichoplax over, it flips itself back because it knows top from bottom. It's about as simple as you can be and be a multicellular organism. And you notice it has 0.04 picograms of DNA as a haploid amount. This is a Homo sapiens. I trust you recognize our friend here. We have about 3.5 picograms of DNA, so we're talking about roughly 100 times as much as trichoplax, the walking pancake. But this is a lungfish, and it has 50 times as much as we do. Lungfish are very ancient. We see fossils of them that go back hundreds of millions of years. I don't quite know why they have 50 times as much DNA as Einstein does, but there you are. This is a less exciting but more detailed representation of this, pointing out that you need to be careful what you use for your information. And this author was pointing out that if all you use are sequenced genomes as your samples, you're going to bias your sample. Typically speaking, scientists are human, and they like to do things the easy way. So when you're going to sequence something, you try and pick the smallest genome for that you can, right? And so in the process of picking genomes to sequence, the first ones in a group are usually picked from the ones that have a fairly small genome. It's only after we've done the easy ones we go on to the hard ones. And this line is showing you the correlation when you look at the easily sequenced organisms with genome size and the number of genes. Out here, you're seeing the rest of the organisms in that evolutionary group, the amphibians and the birds and whatever. And you can see there's a very large variation within a given group on the actual amount of DNA that it has. Same number of genes, roughly speaking, but there's more DNA in there. This is one, I suspect this might not be legible on your handout. Let me see. Yes, it is, if you really look close. Don't worry, I'm not going to ask you to tell me any of them. But I like this one because it has a more complete listing of these things. The protozoa here span well, they're very, very diverse. But this gives you a sense that this is quite a general problem of having varying amounts of DNA for a common kind of organization. There is some relationship between genome size and some biologically interesting properties. So for example, the larger the genome, roughly speaking, the larger the body. And this is showing you curves like that for 2 different groups. This is showing you amount of DNA and body size for flatworms. And this is showing you amount of DNA and body size for insects. And so there is some correlation to body size. This shows up even at the level of the cell. This is showing you for 2 different fishes. This is showing you red blood cells for 2 different kinds of fishes. This is the fighting betta fishes from Thailand, I think it is. And this is our old friend, the lungfish, who has more DNA than Einstein. And you can see that the betta has these little tiny red blood cells and has a small genome. And the lungfish, with its huge genome, has huge cells. And even perhaps a functional consequence. So if we look at the genome size of birds, we see that the smaller the genome, the stronger the flyer. And that seems maybe not like an obvious connection, except that you can have better oxygen exchange with smaller cells. So if you need to have small amounts of DNA to have small red blood cells, then perhaps that's one way of having small enough cells to be a hyperoxygenated bloodstream so you can be a strong flyer. Have a great weekend. I'm happy to answer questions.